Well, thank you, Chloe, for the lovely introduction, and thank you all for joining us. Okay, so this presentation will be basically divided into 4 parts. So first, I will go over a brief introduction on mRNA sequencing. Then I'll talk to you a little bit about our regular, low, and ultra-low input pipelines, followed by our FFP pipeline. And I will finish the presentation talking about our stranded or directional mRNA sequencing pipeline. But first, if you haven't heard about us, we are Novogene, and our goal is basically to be the global leader in genomic services and solutions. We do have offices and laboratories all across the globe. Myself, I work at Novogene USA in our Sacramento office, and we have collaborated with over 4,000 partners worldwide. So please do not hesitate to contact us with your genomics inquiry. We would love to help you to get the best data possible. So without further ado, let's start the presentation. So let me start by giving you a little bit of an introduction about mRNA sequencing. So the first question that we need to ask ourselves is, what is mRNA, right? So RNA is nothing but a single-stranded nucleic acid polymer composed of 4 different nucleotides, A, C, G, and U, and they're joined through a backbone of alternating phosphates and ribose residues. Below you'll find the list of the types of RNAs that we have in eukaryotic cells. They are basically divided into coding RNAs, which are the messenger RNAs that code for proteins, and we have the non-coding RNAs, which are also divided into 2, regulatory non-coding RNAs and housekeeping RNAs. Regulatory non-coding RNAs encompass long non-coding RNAs, small non-coding RNAs, and circular RNAs, whereas the housekeeping RNAs are the tRNAs and rRNAs, which build up the ribosome. If you had a little bit of background in biology before, you have seen this before. It is the central dogma of biology. So extending a little bit on the thought of what is mRNA, we have basically the central dogma of biology. So it states pretty much that DNA is transcribed into mRNA in the nucleus of an eukaryotic cell. It is then edited and transported to the cytosol, where it's translated into protein. When we perform an mRNA sequencing, we are measuring the abundance of this macromolecule here in the middle, which is basically the mRNA. Okay, so now that we understood what is mRNA, so how do we do it, right? So here at Novogene, we use a combination of Illumina and Falcon platforms, which integrates 66 precision devices, combining the entire process of sample extraction, library construction, pooling, and bioinformatic analysis into one single intelligent platform. It really helps us achieve the price that we offer you guys and also the exceptional TATs. Now that we know a little bit of what it is and how we do it, so why would we use that? So eukaryotic mRNA sequencing can answer a variety of scientific questions, from gene expression levels, gene classifications, and gene network and pathways. In summary, the most used analysis by our clients are gene expression, quantification, functional analysis of differentially expressed genes, gene structural analysis, differential expression among groups, and identification of novel transcripts. Just as an example, in a little bit more depth, we can mention, for example, in oncology, the study of fusion genes and analysis in pathological mechanisms, gene expression variation in drug-treated samples, and lastly, disease-related gene analysis in personal health. All, of course, these are just examples, so the list keeps going. So if you're interested in learning more, please just visit our website or email us, and we would love to answer your questions. Okay, the second part of this presentation is basically going to be on regular, low, and ultra-low input eukaryotic mRNA sequencing. Shall we? So to kick this off, I would like to go over with you the different requirements that each of these pipelines have. So for the ultra-low input, we need at least 10 picograms of total RNA with an RNA integrity number of at least 5.8. For the low input pipeline, we need at least 100 nanograms of total RNA with a RIN number of at least 4. And for the regular pipeline, we need at least 400 nanograms of total RNA with a RIN of 4. Obviously, the lack of contamination in all of these pipelines is a must if we want to ensure a good quality data. So here's a quick scheme of how the mRNA-seq pipeline in our company works. So we start by receiving the total RNA from you. Then we perform our sample quality control, followed by library construction, library quality control, sequencing data quality control, and BI analysis. You can take a look and you can see that every single step of the way has a quality control check. So this provides us with a really good insight if the data is not as good as we want it to be, we can exactly pinpoint where the error happened and we can fix it pretty fast. So let's start by going over the sample quality control. So for sample quality control, we use the Agilent 2100 for qualitative and quantitative detection. So on the right-hand side of this figure, you can see examples of samples with different RNA integrity numbers values. So the top one being the best one with an RNA integrity number of 10, and the bottom one being the worst one with an RNA integrity number of 3. Next in line, you have the library construction. So for the library construction for the ultra-low input, total RNA is reverse transcribed into the first strand of cDNA. Double-stranded cDNA is then synthesized by LD-PCR amplification, followed by the purification with M-pure beads and quantification with qubit. The cDNA samples are fragmented and repaired, A-tailed, and ligated with adapters, and after size selection and PCR enrichment, pretty much the library is ready for sequencing. So the only difference in these pipelines, when I'm talking about these pipelines, I'm referring to ultra low, low, and regular, is pretty much the library construction. So on the other hand, for regular and low input mRNA sequencing, messenger RNA is purified from total RNA, the total RNA that we received from you, using a poly-T oligoattached magnetic beads. Once we have only mRNA, it is then fragmented, first strand of cDNA synthesized using random hexamer primers, followed by the second strand of cDNA synthesis. After that, it is end repaired, A-tailing, adapter ligation, size selection, amplification, and purification of the RNA. The library is ready for sequencing. Again, just to keep in mind, the library construction is the only difference in the pipeline between regular, low, and ultra-low input. So in our library quality control, which is the next step in the workflow, the resulting libraries are checked with qubit and real-time qPCR for quantification and size distribution. The qualified libraries will be put together and sequenced on an Illumina NovaSeq 6000 with the strategy PE150, which means pair end 150, and we usually recommend at least 20 million reads per sample. After the sequencing is done, we perform our data quality control. So in this step, we analyze the data through its error rate distribution, GC content distribution, and then we perform some data filtering, where we filter out low quality reads and reads containing adapters. All the downstream bioinformatic analysis performed at Novogene is done with clean reads, which usually for a sample that has passed our standards of quality control is above 80%, but most of them is over 90%. So we're really proud of that good quality data. For our bioinformatic analysis, we have several options, but the most common are quantification analysis, either for human and mouse or species with reference, as well as our quantification analysis, as well as our standard analysis for human and mouse or species with reference. But don't worry, if the species that you're interested in does not have a reference genome, we also offer a variety of de novo pipelines for us to get that done for you. So since our human and mouse standard analysis are the most sought after, let's go over together the workflow. It all starts with the raw data that was acquired from the sequencing reaction, right? So this raw data is then qualified and filtered by error rate distribution and GC content distribution. Once we have only clean reads, we will then map it to the reference genome. After this is accomplished, our workflow branches out either to our structure analysis, where we analyze alternative splicing, single nucleotide polymorphisms, insertions, deletions, and fusion genes, or gene expression quantification, which will culminate into the differential expression analysis of genes and their function analysis. So basically, after the data was mapped to the reference genome, we separate into two different analyses: structure analysis and gene quantification analysis. If any of these analyses seem interesting to you, just please contact us. We have demo reports as examples, and the data is really ready for you to publish right after it is all accomplished. So in the third part of this presentation, I will go over FFPE samples and how we perform our RNA-seq analysis on them. So here at Novogene, by having a standardized pipeline for FFPE samples, it allows us to use the power of next generation sequencing to measure gene expression patterns in typical clinical samples. So our pipeline is an amalgamation of the TrueSeq RNA exome library prep kit with our in-house bioinformatic pipeline. This combination provides us a robust and reliable transcriptome analysis of RNA isolated from FFPE samples or even other low-quality clinical samples. The FFPE RNA-seq workflow is pretty similar to our eukaryotic mRNA-seq pipeline. It goes from sample processing to library construction to our bioinformatic analysis and finally to our final report. So if you want to send us FFPE samples, how much of those do you need to send us? So if you intend on sending FFPE samples, you should send us at least 10 4-microgram sessions, and each one of them with a tissue area of at least 25 micrometers squared and a tumor content of at least 20%. If you want to send us extracted RNA, we need at least 200 nanograms of purified RNA and at least 20 microliters of any buffer that maintains the quality of this extracted RNA. We prefer if this RNA was previously qualified and quantified by qubit and has a 216 to 280 value of above 2, of course. We don't like degradation, and we also do not like DNA contamination, just to make our life a little bit easier. But don't worry, if your data is not, if the sample is not super good quality, we can provide you some purification steps. In this case, where the sample is degraded, we recommend 40 million reads per sample just to account for the possible degradation. And if the sample passes our quality control, we do guarantee the quality of the data. Lastly, on the FFPE workflow, we have our bioinformatic analysis, right? So here we start with the raw data and we perform the same data quality control as the previous pipelines, filtering out low quality data and moving forward only with high quality data. After that, we map the data to the reference genome and we branch out to our advanced analysis, where we do our structural analysis of splicing, single nucleotide polymorphisms, and insertions and deletions and fusion genes, or we branch out to our standard analysis, where we perform gene expression quantification and analysis. So please keep in mind that we also have customized analysis for all of these pipelines. And if you can't find the answer to your scientific question in this slide, don't worry. Talk to us and we're going to figure it out for you. Lastly, but not least, in this presentation, I'm going to go real quickly over our directional or stranded mRNA-seq pipeline. So usually, the non-stranded mRNA-seq pipeline is sufficient for measuring gene expression in organisms. And the benefits of this approach is low cost and high throughput, whereas stranded mRNA sequencing is strongly recommended when the information on the directionality of the transcript is required. It is usually really good for studies of sense and antisense transcripts. Okay, so in summary, compared with regular mRNA sequencing, stranded-specific mRNA-seq can retain the stranded specificity of the origin sample in each transcript by incorporating UTPs instead of TTPs during the synthesis of the second strand of cDNA in the library preparation protocol. With Novogene's stranded mRNA-seq for eukaryotes, it becomes possible to accurately quantify gene expression levels for overlapping genes that are transcribed from opposite strands of the same DNA locus, besides making it easier for us to perform downstream analysis such as genome annotation and novel transcripts detection. The workflow for a directional or stranded pipeline is the same as a regular mRNA-seq pipeline with one tiny variation during library prep that I just mentioned to you guys. So we still ask for at least 400 nanograms of sample of total RNA in good quality. We're going to perform the sequencing on our NovaSeq 6000 machines using the PE150 strategy, and we recommend, in this case, 20 million reads per sample. The bioinformatic analysis also follows the same pattern as a regular mRNA-seq pipeline. So it's starting with broad data, going to data quality control, and mapping to the reference genome, followed by our gene expression analysis and comparisons and structural analysis. So to sum it all up, here in Novogene, we provide you with high-quality mRNA-seq data for regular samples, low-input samples, ultra-low input samples, and highly degraded samples. Also, if you're interested in quantifying overlapping genes that are transcribed from opposite strands of the same gene locus, we can also help you. So really, I cannot emphasize this enough. We are here to help you solve your genomics questions, so please don't hesitate to contact us if you have any. This is it. Thank you.